After medical school, we trained in residency in pathology, and I started out at Columbia University in New York, which trains academic professors of pathology, and they become specialized in one narrow corner of practice. I wanted to do basic diagnostic pathology for doctors and patients, and I wanted to cover a broad area of diagnostic medicine. So I transferred to NYU Bellevue, which had a special training program in just that area. So I gave up the academic professor type track, and I went for the patient service community hospital track, which involves learning the skills in diagnosis with the microscope. So we analyze surgical specimens and do autopsies. We gain proficiency in blood banking and clinical microbiology and hematology, chemistry, blood serology, and all of the things that a hospital pathologist is called upon to do. A pathologist is really a consultant for every one of the specialists, so we have to speak the language of all the medical specialties. And we have to know diagnostically what their problems are and help them solve their problems so that they can take care of their patients. And so we're a patient-oriented but broad-based specialty. And that's why so many of those skills helped me in my research along the way, because I learned a lot about making slides and studying under the microscope and looking for antibodies and making reagents and doing controls and computer things and programming and blood studies, blood smears, blood banking. All that stuff came together under one package. And then the phone calls would come in during the day, and the specimen would come down from the operating room, and I would diagnose from morning until night. And it would be benign, malignant, in between, and all the diagnoses that I wrote or spoke about were expected to be 100% correct. If a pathologist makes a mistake, and we are human, we can make mistakes, everybody gets sued, and the patient suffers. So our error rate is not tolerated, even at a fraction of a percent. If you're an internist and you're 80% right and 20% wrong, you are a wonderful, wonderful internist. If you're a pathologist and you're wrong 20% of the time, you're unemployed. I wanted to do, as a special area within my broad area of pathology practice, an area called tissue tropisms in infectious disease. So I was microbiology oriented from the very, very beginning. And tissue tropisms mean that the bug seeks out an area of the body to cause trouble in. And tropism means that there's an attraction for the bug to the tissue, or the tissue binds to the bug in preference to other areas of the body. And so I was interested in tissue tropisms in infectious disease. Viruses have them, bacteria have them, fungus has them. You won't find viral infections of the toenail, but you'll find fungal infections in the toenail, for example. So podiatrists make a nice living taking care of fungus in the toes. These are examples of tissue tropisms in infectious disease. Now, syphilis, of course, was the ultimate target for study of a broad area of organ system troubles due to one organism, and that's the syphilis spirochete, which is a cousin, just by coincidence, to the Lyme disease spirochete. It's not transmitted in the same way the ticks transmit with tick bite, the Borrelia organisms in Lyme disease. And of course, there's a sexual connection with transmission of syphilis. Syphilis could get just about every target in the body that you can think of. And so over hundreds of years, 500 years, all these things were compiled and collected in a textbook. And there was a masterful work in 1945 called the Stokes Textbook of Syphilis. And it had so many complicating examples of ways syphilis could present. And people had to learn it by heart because syphilis was a public health problem, big time in 1945. The physicians used to call the textbooks the green monster because it had so much information in it and so much to learn and so many exceptions to the rule, and it was a challenge. In 1981, I was a young pathologist beginning my practice in Southampton, New York, and I was doing my work, and I was approached by Dr. Jorge Benach, who now is a world authority on many, many things connected with Borrelia and Lyme disease, and also head of the Biodefense Laboratory and a full university professor at Stony Brook. But at that time, he was a young investigator, and he brought his colleagues out to Southampton because he knew that we were knee-deep in ticks. We were close to Shelter Island, where the cause of the infection from a tick that lived on Shelter Island had been recovered and captured by Dr. Benach and sent out to Rocky Mountain Lab. And he wanted me to interest the medical staff in looking for cases of Lyme disease in each of the specialties. Now, that was an open invitation for me to open up the green monster, wasn't it? So I did a public relations campaign to interest all of the physicians at Southampton Hospital, and all of them were interested, and all of them found examples of the disease in their specialty. And we presented a full-day symposium in 1987. We had Dr. Willy Bergdorfer there as a keynote speaker. And we had many other visiting professors, including Dr. Benach as a young professor. And we did Lyme disease from A to Z. And we had examples of Lyme disease in each of the clinical specialties in the textbook of medicine. Dr. Paul Lavoie was alive at the time. He said, you know, whether you realize it or not, we've just gone through the whole textbook of medicine. We just opened up the page and we flipped, and each page has an example of Lyme disease in a specialty area. We've just done that today. There are a couple of reasons that I am a career Alzheimer Lyme researcher. The first is that my grandfather, who was a full professor at Harvard Medical School, died at a relatively young age, 67. I'm 65 now. So he died after a long illness with dementia. And he had an autopsy at Harvard Medical School, and they found that his dementia was related to degenerative disease. And we thought originally it might be Alzheimer's, but it turned out to be Lewy body disease, which is sort of a first cousin of Alzheimer's disease. And so he died of Lewy body disease. And his whole life was wrecked. And my mother would go into the pantry after he passed away, and she would cry every day for a long time. And you know, it made a big impact on me. I wanted to do something to see if I could find more about dementia. And in syphilis, there is a very, very important example, and that is the dementia called general paresis. And general paresis is very, very late stage syphilis of the nervous system causing dementia. Al Capone had it. Other people had it. And it is syphilis of the nervous system which causes dementia. And when I went to a conference in 1985 in Vienna on Lyme disease, they mentioned stages of Lyme disease in the nervous system: primary, secondary, tertiary. Now, if you've read the Green Monster textbook for syphilis, you know that primary syphilis is early syphilis, secondary syphilis is in between, tertiary syphilis is way, way out from the time of infection, and the real example of tertiary syphilis is brain deterioration or dementia, general paresis. So when they said tertiary and then they said Lyme, my mind immediately did an electrical lightning bolt and arced to tertiary syphilis is dementia. Why can't there be tertiary Lyme disease with dementia? And if there is such a thing, Alzheimer's is most likely the target. And so I spent some time working with Alzheimer brain tissue, which I received from brain banks. And I started with George Glenner's brain bank in University of California, San Diego. I received by air freight, Federal Express, frozen brains and dry ice, and I put them thawed in culture medium, and I was able to grow the Lyme spirochete from 4 of 4 Alzheimer brains from George Glenner's Alzheimer Brain Bank at the University of California, San Diego. And I sent this report off to the Journal of the American Medical Association because I was excited about it. And they were reluctant to publish it because they thought it would provoke a lot of public concern and a lot of anxiety. And so we got a rejection letter of that. Even though I had pictures of the spirochetes, pictures of the confirmation with the antibodies showing they were Lyme spirochetes, and I had pictures, galore of what I had done. So we worked out a compromise, and we did a smaller letter to the editor. It's not a full article, but it gets published. They let me publish 4 of my pictures, and it was entitled Borrelia in the Brains of Patients Dying with Dementia. And the date of that publication was 1986. That's 1 year after I got the lightning bolt idea from Vienna, Austria, about the possibility that Alzheimer's might produce some Lyme spirochetes if we allowed the brain tissue to grow in the right condition. Since that time, my techniques have become more and more sophisticated, and I've gotten past the culturing of brain. I've gotten into the molecular and the DNA aspects and stained Alzheimer brain tissue with special reagents to show the spirochetes at the site of the Alzheimer injury. In Alzheimer disease, there is normal brain, and then there are areas that are like round, burned out areas that are destroyed brain tissue. So you have sort of a checkerboard. You have normal, and you have disease, and then normal and disease. And the areas of disease can range from very small round circles to much larger round circles. These are called plaques. Plaques are area where the brain has been destroyed. And I focused on the plaques, and I focused on some of the other areas of Alzheimer's, one area called granular degeneration. And I found in each of those areas of study, using the most sophisticated DNA probes, that Borrelia DNA is present in the areas of Alzheimer injury in some brains from the Harvard University Brain Bank, where I now receive specimens. I'm an affiliated investigator at the Harvard Brain Bank, so I can receive any number of specimens from them, and I continue my study. I'm doing my work now at the University of New Haven in Dr. Eva Shoppe's laboratory. And we had some great results yesterday, which I'll tell you about if we have some time. So the work continues, and we are finishing a case study from a German physician who had Alzheimer's disease, had high levels of antibodies to Lyme spirochetes, actually 3 different strains in his spinal fluid. Studied his brain, bingo, positive. We found the Alzheimer areas showed Lyme spirochetes. Remember, I'm a pathologist. My area of expertise is diagnosis of disease under the microscope. And therefore, I'm a quality control officer, and I look at tissues and I diagnose if I can, or I send to experts who can help me diagnose, and ultimately a pathologist is the one who diagnoses the disease. Now, in pathology, when we find an area of tissue injury, and we can name that with a disease name, a label that we understand, like strep throat, and in the strep throat tissue we find streptococcus bacteria, then we firmly believe that the streptococcus bacteria cause the strep throat. And if we look in the appendix that has been ruptured and we look under the microscope and we find lots of bacteria, we believe that the pus in the appendix came from bacteria that infected the appendix. And so the bacteria there were the cause of the disease. In general paresis, this work was very difficult because it was really one of the last areas to be brought under the firmly associated with syphilis category. And that work was done at the Rockefeller University in New York by Dr. Hideo Noguchi. And his work in the Rockefeller proved, after many, many years of frustration with histologic stains, that the syphilis spirochete was present under the microscope in the brain of the general paresis patient. And that was a major, major step forward. And that brought the last of the symptoms, that dementia symptom, general paresis, into the firm community of diseases that are caused by syphilis. Now, finding the spirochete at the site of the Alzheimer injury is not any different from finding the strep at the site of the strep throat, or finding the bacteria at the site of the ruptured appendix, or finding the syphilis spirochete at the site of the general paresis. There is no difference. The spirochetes are not waiting for a bus. They are not reading the New York Times. They are there to do injury, just like other injury-producing microbes of many, many species are there to do injury when they're in certain parts of the body. Now, bacteria can be our friends too, and it's estimated that of all the life in a body, 90% by weight of the human body is bacteria. And those bacteria live in biofilm communities. And our cells are a regular small player. And most of our body is based on healthy bacteria living in healthy communities and doing good things for us, making vitamins and promoting the general welfare of the body. So bacteria can be our friends, but bacteria can also be our enemies. All right, Borrelia is a spirochete, and the spirochete is controlled by DNA, and the DNA then produces proteins. So when test materials are manufactured, the Borrelia are grown in huge tanks the size of this table, and then they are made into a pellet, like, packed tight, and then they're digested, and either the proteins are extracted or the DNA is extracted. And then the proteins are then placed in paper carriers, and they're used for diagnostic testing. So the ELISA test has the proteins of the Borrelia on a paper disc, and the Western blot has the proteins spread out in the ladder array on a paper strip. We have all of our tests based on one strain of bacteria, and that strain is the B31 strain of Borrelia, which came from one tick, and it came from the growth of that tick's gut spirochetes in a test tube in Rocky Mountain Lab. But it wasn't pure because Dr. Barber, who did that work, wanted to be absolutely sure there were no other bacteria mixed in with it. So what he did is he called into action a procedure called limiting dilution cloning. And that meant that if you have a test tube with 10 million bacteria and you dilute it and dilute it and dilute it and dilute it and dilute it, there will come a time after a series of dilutions where there's only one spirochete left in the entire test tube. And he did that. He cloned this B31 strain to one spirochete in a test tube, and then allowed that to take off and grow and grow and grow and become a huge cell line, which now is the basis for all of our USA test kits. Now think about it. One spirochete and one lineage, one set of proteins, one DNA is going to be the ruler for diagnosing any and all of the bacterial spirochetal Borrelia infections in the United States. We now know there are at least 100 different genotypes of Borrelia burgdorferi, USA type. And we're working with one test kit from one spirochete with one set of proteins, one set of DNA molecules, right? And that's the ruler for all of the other 99 strains that we know about in the United States. And that spirochete never saw a human body. That came from a tick. So we don't even know whether that spirochete would have been able to make it into the human body and cause disease in a human in a real case. Came from a tick gut. So there's a lot of flaws in relying and betting the ranch on one spirochete and it's all its progeny and one test kit, because we have many strains of Borrelia in the United States and other strains around the world, and those do not react very well with our USA test kit. So if you come from Europe and you have a European strain and you have Lyme disease of the European strain type and you get tested with a USA test kit, you will get a negative result. That's scary. But if you go back to Europe and have your blood tested with European manufactured test kits, you're likely to get the positive result that you deserve. So betting the ranch on one is a flawed concept. In Europe, there are at least 3 strains that are in play, and the 3 strains are Borrelia afzelii, A-F-Z-E-L-I-I-I, or 2 I's, Garinii, G-A-R-I-N-I-I. These are Latin words, and please excuse my Latin pronunciation. I pronounce Latin as well as Mayor Bloomberg pronounces Spanish. So it's not a pretty picture, but I do the best I can. And then the Burgdorferi variant in Europe may be somewhat different from the Burgdorferi variant here in the United States. For each of those strains in Europe, they have manufactured test kits which are derived from the strain. So instead of having one test kit, they have at least 3. And you run the blood against Afzelii, Garinii, and European Borrelia, other. And therefore you have a wider net and a better chance of finding out whether the infection is due to one of these Borrelia or whether it's due to all 3 of them. You know, we recently completed an autopsy study of a German physician who in his spinal fluid had evidence by Western blot of all 3 Borrelia strains simultaneously infecting his brain by Western blot. He had like 14 bands. The CDC likes it if you have more than 5 bands. He had 14 and up to 20 for some of the strains. So he definitely had multiple infection with European strains. There's another example of a person who was a Stony Brook patient but had acquired Lyme disease while camping in Europe, bitten by a European tick. Came back to the U.S. At Stony Brook, it's a great center for Lyme disease. Many, many professors and luminaries in Lyme science are at Stony Brook on faculty. And they wrote a paper called Relapsing Seronegative Chronic Lyme Borreliosis of the Nervous System. And the problem was they were looking for European evidence with USA test kits. And there were some other problems too that made it complicated. But the blood tests were negative. And that's because you were dealing with a European strain in a US test lab. We have world travel. We have people who travel, and we have people in military service. We have people stationed in Germany. Germany has lots of hot spots. The Black Forest is one of the hottest spots for Lyme disease in Europe. Our military personnel who acquire infection on maneuvers in the trenches in Germany may come back with that infection and then depend on USA test kits to diagnose a European infection. They don't get good success with that. So it is very complicated. You have to be a member of a global community, and you're dealing with many possible strains because of air travel, military service, and exposure to strains other than the B31, which came from the tick that never saw a human. That is the basis for the entire test kit system in these United States. Isn't that a remarkable scenario? Remarkable. Military intelligence. Virulence is the ability of a microbe to cause disease in a host. And there are many ways of measuring virulence. You can see measurements based on how many, by number count, you have to inject into the body of the victim to get the established infection going. So you can do that by number. You can do it by what is the ultimate outcome of the infection. Do the patients live or die? So you can do death counts versus survivor counts and get a feeling for virulence. And there are other ways you can look at virulence, but those are simplified ways. And each strain of Borrelia, each named strain, and each of the genotype strains has differences at the level of DNA which depart from the B31 DNA, which is our test kit organism DNA. And that is expected in microbes in the wild. They're out there interacting with other species, interacting with ticks. Their DNA is reproducing, and during reproduction, things can happen. You can have point mutations, you can have other mutations. You can have things where transfer from DNA of one strain of Borrelia is moved over to another strain, and the strain that acquires the Borrelia that's moved over to it from its neighboring Borrelia strain becomes more aggressive and different. And there are what we call fluxes. Fluxes are, it's a physics term, I guess, and it has to do with magnetism and shifting of the North Pole to the South Pole, the North Pole to the South Pole. There's fluxes in DNA too. And if you can imagine DNA undergoing activities which change it over a period of time, it's a molecule in flux. And molecules in flux are not unknown to us as general members of the community. Influenza virus does it. DNA in flux is something that happens at the microbial level, at the viral level, and it's just a manifestation of life on Earth. The DNA is not static.